In today's video, we will show an immunofluorescence protocol for C-Fos. C-Fos is an immediate early gene that is expressed upon neuronal activation, making it a good marker when studying neuronal activity using techniques like immunofluorescence. Today, we will be going over the steps for an immunofluorescence protocol to detect C-Fos in neurons. For day 1 of the C-Fos immunofluorescence protocol, begin with retrieving your desired tissue samples. We will be using 50 micron coronal sections from a rat brain. Next, we will be performing 3 5-minute PBS washes. Obtain a 12-well plate and add 1x PBS to all wells. We are using plastic well inserts to facilitate the transfer of tissue between wells. Add your tissue to the wells of PBS and place on a shaking belly dancer for 5 minutes. Move the tissue to the next well and repeat 2 more PBS washes. Next, create your blocking solution consisting of 3% normal goat serum and 0.5% Triton X100 and 1x PBS. The purpose of blocking is to minimize background staining, as the goat serum will bind to non-specific substrates. Triton X100 is a detergent that increases the permeability of cells. Incubate your tissue samples in blocking solution for 30 minutes at room temperature on a shaking belly dancer. Prepare your primary antibody solution with a 1 in 500 dilution of C-Fos primary antibody diluted in blocking solution. Pipette 1 milliliter of the primary solution into each well and transfer the tissue out of the plastic well inserts into the solution. Parafilm the well plate and leave it shaking on a belly dancer at 4 degrees Celsius for 24 hours. During this incubation period, the antibody will specifically bind to C-Fos. For day 2 of the C-Fos immunofluorescence protocol, retrieve your samples from the fridge and conduct 3 5-minute washes in 0.5% Triton X100 and 1x PBS to remove any unbound antibody. Prepare your secondary antibody solution using a 1 in 250 dilution of a biotinylated secondary in blocking solution. Pipette 1 milliliter into each well and transfer the tissue to the wells. Incubate at room temperature on a shaking belly dancer for 2 hours. After the secondary incubation is completed, perform 3 5-minute PBS washes to remove any unbound antibody. Next, place your tissue into blocking solution, once again consisting of 3% normal goat serum and 0.5% Triton X100 and 1x PBS for 30 minutes. Prepare a solution with a 1 in 500 dilution of streptavidin conjugated to cyanine 3 in blocking solution. Pipette 1 milliliter of this solution into each well and transfer your tissue into solution. Note that cyanine 3 is a fluorescent dye and is light sensitive. Therefore, ensure that this and any following incubations are performed in the dark to help minimize the potential for photobleaching. This can be done by wrapping foil around the well plate. Incubate in streptavidin cyanine 3 solution for 1 hour at room temperature on a shaking belly dancer. Next, perform 3 5-minute PBS washes. Now you can mount your tissue onto microscope slides. Next, perform a slide-mounted hook stain using 1 in 1000 hooks diluted in 1x PBS. Hooks is used as a counter stain as it binds to the minor grooves of DNA and therefore fluorescently labels cell nuclei. Let the hooks sit on the slides for 15 minutes in the dark. Remove the hook solution from the slides and perform 3 5-minute PBS washes on your slides. Wait for the slides to partially dry and then cover slip with Fluoramount G mounting medium. Leave your slides covered in the dark overnight to dry and then image using fluorescent microscopy.